get active here in the forum. Well, as always, or as usual, I will start with a bunch of announcements. One is that today at noon upstairs in CST 300, Penny Smith will be guiding a conversation about the ethics of human genome editing. A reminder that the quiz 1, will be available tomorrow. It's due on Wednesday night, 11:30 p.m. Closed book. No notes, no books, no electronics. Leave them far away from where you're sitting when you're taking the quiz to avoid the temptation to look things up. Pick up the quiz at the CST desk. Take it. Turn it back in to the CST desk. It doesn't leave the building. Do it right here. Quiz vote. If you would like access to the AMP lab to do AMP Revealed in preparation for the quiz, that can be arranged. Contact me or any of the TAs, and we all have access to that room. We can let you in, and we'll power up the computer you need to run AMP Revealed, and you can review AMP Revealed. You can also pick up the CD-ROM here at the CST desk and run it in any computer that has a CD-ROM drive. Yes?
If we wanted to do that, does it have to be during the week, or should we try to do it during the weekend?
You can try to do it during the weekend. You just have to make an arrangement with either me or the TA. So, if you can persuade one of the TAs to come in and let you in, or if maybe they're already here, that's fine. I'll be here on Sunday, so I can, you can work on this on Sunday instead of, or, go back and forth between the brain and the AMP Revealed on Sunday. So we can do access. We just have to make arrangements. I can't leave the door open. I just can't leave the room open. It's against the rules. But I can give you as much access as you ask for. Brain dissection on Sunday.
Do we have to ask public safety for access to the lab for this class?
Say again?
We can't ask public safety for access.
I would rather not go that route. That would be a backup. If necessary, you could contact public safety. I think they would let you in if I alert them that I don't know how well they communicate with each other.
But I meant to give us access for the whole semester for the lab.
Oh, I could do that. Not crazy about that because the rule for having a cadaver is that it be behind 3 locks. So that is the rule. At least that's what I've been told the rule is. So the hallway door is locked, the door to the room is locked, and the door of the cadaver gurney is locked. So there's our 3 locks. If I mess up, if I say the room is unlocked or give wide access to it, I'm a little nervous about jeopardizing my compliance with the rule. I could find out. I could try to find out. Problem sets available means that I have your graded problem sets, so I can hand it out. I can hand it back after class. That's what this was in mind for me to tell you that. So if you want your problem set, your graded problem set, just stay for a few minutes after class. And because of a basketball game that didn't happen yesterday but is happening tonight, there's a few students who who are in the Thursday evening lab who have been moved temporarily to the Tuesday lab. It means that the Thursday evening lab is pretty sparse, it's pretty, has vacancies, and the Tuesday labs, both of them, the 1 to 3 and the 3 to 5 shift, are relatively crowded. So if you are in either of the Tuesday lab sections and you can come tonight, please do. It'll give you that much more time to prepare for the quiz, and it's available. The room, there is at least 4 slots available. Okay, so questions about any of that? Yes?
The material for the quiz, it has more of what we learn in lab?
Yes.
Or does it have questions for the
It's It's anatomy,
So,
almost 90, it's almost all anatomy. So your resources for the quiz are the coloring book assignments, the A&P revealed assignments from lab, your worksheets that you have from lab, and the list, the checklist. The quiz will not cover physiological material. Other questions? Yes, David?
Is there a way that we could get our problem sets back specifically so we can see where the mistakes were made?
Yeah, I'm gonna give you back the problem sets after class today.
Okay.
Okay, so here we are. Studying action potentials, well, synaptic potentials, membrane potentials, and action. So remember, what we were focusing on before, on Tuesday, was the resting membrane potential of this neuron. We did that and we worked with that on problem set material. Then how neurotransmitters and neurotransmitter receptors create EPSPs or IPSPs in this location. How they summate, the thousands of them that are happening all the time, summate, either positively or negatively, that is to say, add up or subtract off from each other, to create an overall depolarization of this neuron that can depolarize to the point of reaching threshold, where then this neuron would fire one or a series of action potentials. The axon hillock is where that integration, the soma and the hillock are the most important parts for that integration of EPSPs, excitatory postsynaptic potentials, and IPSPs, inhibitory postsynaptic potentials. The axon hillock, the axon initial zone, the spike initiation zone, is where threshold really happens. I mean, if this region reaches threshold, then it fires an action potential which propagates along the axon. The threshold here is set by the properties of the ion channels here. One thing that is true about the ion channels that are expressed here at the axon hillock, or the spike initiation zone, is that there is a high expression of voltage-gated sodium channels. Those channels responsible for the upstroke of the action potential. And that's also true at these nodes of Ranvier. High expression of voltage-gated sodium channels. So now we're transitioning from the EPSPs, IPSPs, integration mechanism, to the, and you know about the action potential, you know, the role of voltage-gated sodium channels, the role of voltage-gated potassium channels, how they contribute to the upstroke and then the repolarization and the hyperpolarization. That's what you know. And now what we're going to do is cover how we get propagation. Wait a second. I'm in the wrong slide. Starts with the first one, same first one. Okay. Glitch. So now let's talk about propagation. So this is a cartoon of a membrane of an axon. So we have lipids and channels. This cut-through donut blob model of an ion channel. Outside, inside. This is supposed to cartoon a voltage-gated sodium channel. This is cartooning a voltage-gated sodium channel in the open configuration. And none of these are illustrating the voltage-gated sodium channel in the closed configuration. But you can imagine that if we pinch off this constriction, it would be closed. What opened this voltage-gated sodium channel? Depolarization from upstream somewhere that we're not seeing. So this voltage-gated sodium channel has opened. What ion is carrying this positive charge? Sodium. Positive charge flows in and diffuses in both directions. Diffusion just goes, it doesn't have a directionality to it. It diffuses, and in fact, what diffuses in the cytosol is not just sodium, but the potassium that's there too, because this sodium ion is going to electrostatically repel any potassium ions that are near it. And so potassium is going to also diffuse away from this site because of the local positive charge density here. And in fact, chloride will flow in this direction, but both of those flows, potassium moving this way and chloride moving this way, is equivalent to positive charges diffusing away from the site where they were sort of injected by the voltage-gated sodium channel. Okay? So diffusion of charge, diffusion of charge. The point here is that this channel, I guess these are closed channels. This gate here, this doorway here, is supposed to represent a closed channel. It's not very accurate. So this channel is closed because it has not yet depolarized. But this charge here is moving in this direction. It is it is neutralizing some of the negative charges that are surrounding this channel. And so you can see this gate is starting to open. Charge is diffusing. Yes?
Would you say it's almost like a cascade that the channel opens and sodium come in and depolarize other?
Yeah, it's like, yes, it is. It's a cascade in the sense that it's a whole series of events. It's not actually amplifying, but it is like a kinase cascade can be amplifying. But I think you could call it a cascade. So just remember, injection of charge, and it diffuses. So this channel has now received enough depolarization open. Charge flows in through this channel. Charge diffuses in both directions, just like I said. So you can predict what's going to happen here. Now let me tell you what happens here. Remember that voltage-gated sodium channels, after they open, they inactivate. This little ball moving into the pore is supposed to represent the inactivated channel. So now this cartoon is illustrating the fact that there are two ways the channels can be non-conducting. They can be closed because the activation gate is closed, or they can be inactivated because this inactivation particle, piece of the protein, has moved into place to plug the open channel. This channel is, the activation gate is open, but the inactivation particle, piece of the protein, has plugged it up. So this is the inactivated state, and this is a closed state. You see the inactivated, it's not inactivated, it's closed. So I'm going to ask you to be, from now on the problem set, I did not ask you to be precise about this language because I hadn't emphasized it yet. I allowed you to say that voltage-gated sodium channels were closed during the after hyperpolarization, but in fact, they are inactivated. Voltage-gated sodium channels open in response to depolarization and then inactivate. Remember, we are focusing on both changes in space, in location, and in time. This cartoon is a fraction of a millisecond after this cartoon. So now, the channels that were responsible for the action potential here are inactivated. The channels that were responsible for this action potential here are open. Now they're open. And you can predict what's going to happen next. This injection of charge is going to depolarize this part of the membrane. So let's talk about what this inactivation, what happens because of this inactivation. Because of inactivation, there is a period of time during which the action potential cannot happen. So let me go back to the previous slide. If this channel were suddenly depolarized, in fact, it's receiving depolarization from this channel, but it is not opening because channels that are inactivated cannot open. Closed channels can be opened by depolarization, inactivated channels cannot. So there is a period of time after the action potential has been at a place during which the action potential cannot happen because the channels there are all inactivated. And that's called the refractory period. So now we're looking at voltage as a function of time at one location. We're looking at voltage as a function of location at various times. And here we're looking at voltage as a function of time at one location. This is the action potential at one location. And we've talked about the channels that are responsible for this and for this and for this. But now I'm turning it into the language of refractoriness. When the channels that are responsible for the action potential are doing the action potential, the membrane is called absolutely refractory. No matter how big a depolarization we injected at this point in time, it could never fire another action potential because it's in the middle of firing the one that it's firing. So that makes sense. You can't be firing two action potentials on top of each other because of the all or none properties of the action potential. It's all going. It's giving it all it's got. So we could hammer this with a big external depolarization right here, and nothing would happen because it's absolutely refractory. It's in the middle of its action potential. It's doing its all. For a period of time after an action potential, the membrane or the cell is called relatively refractory. So remember, what is happening to the voltage-gated sodium channels? So I just said that here, most of the voltage-gated sodium channels are inactivated. But from here to here, most of the voltage-gated sodium channels are inactivated. But what's happening to them? What's happening to the voltage-gated sodium channels during the relative refractory period?
They're sort of resetting back to being closed.
They are resetting. They are recovering from inactivated back to closed. So an increasing amount of the sodium channels are going from inactivated to closed. And so more and more sodium channels are now becoming available again for another action potential. So if I give it a depolarization here, I might have to give it a bigger depolarization to get an action potential. Threshold is higher here than it is here because some of the voltage-gated sodium channels are still inactivated. So this point right here has a higher threshold, but we could still fire an action potential because we have enough closed, ready-to-go sodium channels so that if we depolarize it here, we would get another action potential. it would require a bigger depolarization because of the elevated threshold here relative to here. So it's relatively refractory. Getting an action potential to fire during the relative refractory period requires a larger depolarization than normal. And you can imagine that right here, we would require an even larger depolarization to get an action potential at this point in time than we would at this point in time, because fewer sodium channels are recovered and available here than here.
So
Great.
the y-axis is still voltage.
This is voltage as a function of time. So now I've defined the relative refractory period. And it, in fact, the combination, the refractory period mixture, defines how fast, how closely spaced together in time, which is frequency, how frequently an action potential can be fired by a given neuron. If a given neuron has properties where the relative refractory period is relatively short, say it has properties where the voltage-gated sodium channels recover very quickly from inactivation, modulated sodium channels. Well, if the voltage-gated sodium channels can recover more quickly than this cartoon, then the relative refractory period would be shorter, and that cell could fire an action potential at a higher frequency than a cell that has a longer relative refractory period. So the longer the refractory period, the lower the frequency, the lower the maximal frequency that a cell could fire action potentials. This cell is going to fire action potentials with a frequency like this, 1 every 5 milliseconds. let's say. If I shorten the refractory periods, I could make a cell that could fire maybe every 2 milliseconds. That would be a higher frequency. Remember, the patterning and the frequency of action potentials is what carries information. So that's what I'm trying to unpack a bit. So, going back to our cartoon of both voltage as a function of space, a function of distance, and as a function of time, we have, so we were here, now this one has inactivated. This one has opened. This one has recovered and could fire another action potential. Diffusion of charge is necessary for this spread, propagating spread of depolarization. In fact, diffusion of charge both intracellularly and extracellularly is required. If we were to somehow block this diffusion, and we can do that experimentally, it never happens physiologically, but if we were to block this diffusion, the system would stop just the same way it would stop if we block this diffusion, because electricity has to go in a circuit. Those of you who have taken physics have learned some of the rules of electrical circuits. So the takeaway message is that diffusion of charge in the cytosol and in the extracellular fluid is required for this propagate and ion channel properties. Voltage-gated activation and voltage-dependent inactivation are responsible for the propagation of action potentials along axons. So you can see here, this is the mechanism that describes how an action potential moves from here to here to here, moves in space as a function of time, moves in distance, along the axon as a function of time. So understanding this cartoon is an important part of understanding propagation. This is just another way of showing the same information. An action potential is happening here and spreading in that direction. After the action potential, the cell is refractory. after is also behind where the action potential is. So refractoriness is both a function of time and space. There's a period of time, and there's a distance in the axon where this piece of membrane is refractory. So you can think of the action potential as spreading along the axon, and the refractory zone behind it is moving as well. So this area here might have a hard time firing another action potential, but this area here is ready to go. The typical velocity for an unmyelinated spread of action potential, the typical velocity for the spread of action potentials in unmyelinated axons that don't have nodes of Ranvier, that don't have myelin, that don't have Schwann cells or oligodendrocytes wrapping them up. There are some that just are plain, like this. A typical speed is about 1 meter per second. So if I touch my finger 1 meter per second means that it takes 1 second for the information to get from here to here. And you know it happens a lot faster than that. So you know that some of the axons in my fingertip must be not unmyelinated. That is to say, they must be myelinated. But some of the fibers in your fingertip, some of the axons in your arm are in fact unmyelinated. The ones that carry pain, for example, are largely unmyelinated. And that's why when you hammer your finger, it might take a full second before you feel the pain. You know the pain's coming because you have sensed it, but okay, wait for it, yeah, there it is. Because of the speed of action potential propagation in the unmyelinated, pain-carrying axons. Yes? Why have we evolved slow and then I'll show you fast propagation? This evolved before fast, so it's an evolutionary artifact, really. And it's not essential that we feel pain as quickly as we feel touch. It's okay for us to wait a second for us to feel pain. And so evolutionarily, these slow axons have been preserved. So let's talk about the factors that influence the speed of propagation of action potentials, the conduction velocity, the velocity at which action potentials are conducted along an axon. So I've just talked about myelination. You know that myelination increases the speed, and we'll talk about that mechanism soon. But another thing that changes the speed is the diameter of the axon. Big axons propagate action potentials faster than small ones. So, and I could explain the biophysics of it, but it's biophysics and I'm not going to. I just want you to know that large diameter axons conduct action potentials at a higher rate, higher speed than low diameter axons. So the smallest diameter unmyelinated axons are the slowest. And that's an example of the ones that carry slow pain. The large diameter myelinated axons carry the fastest. And they're the ones that carry touch and motor information and some fast pain, like from your teeth. You have a very fast response to tooth pain. You have a very slow response to a crush pain in your hand. It's just the different types of axons that carry that different information. So now let's talk about myelination. In mammals, and in many higher organisms, including not lamprey, no, but fish and reptiles and birds and mammals, have a specialization for increasing the velocity of action potential production, and that is myelination. So myelination are the layers of myelin wrapping the axon. This axon is from, is part of the nerve cell. These wrappings of myelin are from Schwann cells or oligodendrocytes. It's from a different cell. Between these wraps of myelin is an area of more exposed axon, and this is where the voltage-gated sodium channels are highly expressed. So in a myelinated axon, it's the same story almost. Well, it's the same story except that, so let me tell the story. So action potential at this node of Ranvier, same story. Voltage-gated sodium channels have opened. Sodium has flowed in. This sodium and this charge can now diffuse much further than it could in the unmyelinated axon because of the insulating properties of the myelin. So instead of having to have sodium channels all along here to propagate this information across this millimeter, we can let diffusion do the work and let the diffusion of charge carry it to the next node of Ranvier, where the voltage-gated sodium channels will be depolarized, open, then inactivate, and allow the action potential to move fast through this zone. This diffusion is relatively slow, but electrodiffusion, electrostatic accelerated diffusion, right? Charges are being repelled by this influx of sodium has made a bunch of positive charges here that is repelling the positive charges and attracting the negative charges, and that process is fast. So this electrostatic diffusion, the electrostatic movement of charge here is fast. And so the action potential can move from node to node to node much more quickly than if we had to move it from one sodium channel to the next, to the next, to the next. So there's actually an interesting video. It's not that interesting, but this, let's just see if this works for you. Can you see this video? So the idea here is this is an unmyelinated axon, and each domino represents the sodium channels needed to do the action potential. So when a domino falls, that's equivalent to an action potential. So you know what happens when this happens. Domino, 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 and it falls. This segment is supposed to represent a myelinated axon where there's a ruler connecting this domino to this domino, which is representing that electrostatic diffusion under the myelinated insulating area. So this is a node of Ranvier, and here's a node of Ranvier, and here's a node of Ranvier, and the movement from the diffusion from here to here is coupled by the ruler or by the electrostatic diffusion in the node of in the myelinated segment of that axon. And so now you can probably already predict what's going to happen. The unmyelinated system moved much slower than the myelinated axon. Does that make sense?
Physically, what does myelination actually do? Are you constricting something, or?
Physically, what myelination does,
How do you have that increased electrostatic interaction?
physically, what myelination does is increases the insulating properties of the axon and allows these, it allows this process to not decay over this distance, but it decays over 1,000 times longer. So the diffusion, so this thing, instead of getting smaller and smaller and smaller over this distance, stays this wide and diffuses much further because of the insulation. This arrow doesn't exist, this arrow doesn't exist, this arrow doesn't exist, this arrow doesn't exist, this arrow doesn't exist because of the insulation. So all of this charge, this arrow doesn't exist, all of this charge gets to diffuse along the axon millimeters instead of microns in distance.
So it's sort of like a conservation of the charge.
Yeah, I guess. I mean, the charge is conserved. It's just that here the charge is being dissipated by the leak of this membrane. And in an axon, in a myelinated axon, it's not being dissipated. Imagine you've got a hot water pipe, and you've got an uninsulated hot water pipe, and a highly insulated hot water pipe. And you let hot water dribble through the hot water pipe. The heat at the end of the pipe is going to disappear if it's not insulated. So myelination speeds up action potential propagation by a factor of 100, which is a lot, 2 orders of magnitude. And so our myelinated axons here in the fingertip are propagating action potentials at 100 meters per second, whereas the pain ones are carrying it at 1 meter per second. Here's a cartoon of the node of Ranvier showing the high expression of voltage-gated sodium channels in the node. A reminder that a nerve that we're going to see in the lab, in the body, a nerve is not one axon. A nerve is thousands of axons. Some of them are big, and some of them are small. Some of them are thickly myelinated, and some of them aren't. It's hard to show. A mixture of big and small, thick and thinly myelinated axons. Could someone lower the curtain back there, just to prevent this? Never mind. It's not that. It's coming from the reflection off the floor. Can't do anything about that. Stand in front of me. A nerve is a bundle of axons. Not one, thousands, hundreds of thousands. So now let's take a break, and we're going to talk about the sequence events after an action potential invades a presynaptic terminal. We'll take a little bit of an early break today, and then get to the release of neurotransmitter. Together. I think I gave you this slide as a handout on the second page, large, so that you can fill in the blanks, fill in the blank. So let's do this together. Well, actually, why don't I let you work in pairs for a couple of minutes, and then let's do this together. So work with a partner and just figure out what are steps 1 through 6.
It's just the neurotransmitters plant crossing the planet and It's hard to tell from the box. I think it's 6. They're just the next activity. I mean, I feel it's considering how you guys are worried about what to write down on the exact
Okay, what is step, let's get started talking about this. What is supposed to be illustrated by step 1? Action potential propagates into the presynaptic terminal. Right? Propagation to the end, the terminal. The terminal is depolarized, big time, by the action potential propagating into it. Okay, what is step 2? Okay, try to use the colors. What is step 2? Voltage-gated calcium channels open. Voltage-gated calcium channels. Voltage-gated calcium channels are very similar to voltage-gated sodium channels in that they respond to depolarization and they open. But they're a little different in the sense that they select for calcium, not for sodium. Okay, what's step 3? Somebody besides me. Oh, is that fine? Shut it. Somebody over here, tell me what happens in step 3. Okay, calcium influx. Calcium influx, increase in the concentration of calcium on the inside. Yes?
Why calcium? Why do we use calcium instead of sodium? Because calcium would-
I mean, why did we evolve calcium as a signaling molecule?
But I just mean like it's doing the same thing. If it needs depolarizing just so certain numbers, does it make good effect with depolarization, or is it just not about
I think the answer is that animals, even plants, evolved calcium signaling before they evolved mechanisms for nervous systems. And so there was this existing system of calcium-dependent changes in properties like calcium-dependent exocytosis and calcium-modulated enzymes and calcium modulation of the mitochondria. Calcium signaling was a module that existed evolutionarily before this. So this is a module that they're just now saying, okay, let's use this module to regulate neurotransmitter release, and let's use something else for this. I'm not an evolutionary biologist, but if I had to guess, I would say, well, that seems plausible. Okay, what's number 4? There's a couple of things in 4. So let's get some terms on the board. Josh.
Release of neurotransmitters.
Okay, what does the calcium do?
It binds to the precinct vesicles.
Yes, okay, so calcium binds to a specific protein. Some of you may know the name of this protein. I mentioned it in self-phys. I didn't ask you to memorize it then, but I am gonna ask you to memorize it now. The name of that protein to which calcium binds is called, anybody? Calcium binds to synaptotag. Synaptotagmin. Conformational change of the V snare complex I'll switch over here now so you can see it over here. So I'm not going to reproduce all that, but I'm going to continue. So we're still at level 4 here, step 4. Conformational change of the V snare complex. V snare, T snare, conformational change, fusion of synaptic vesicles with the surface membrane. Exocytosis. Sorry, go ahead.
Does it do the docking exactly like in self-phys?
It does the docking exactly like in self-phys. This is the calcium, this is the docking and the calcium dependent exocytosis perfected. Pretty close to
Oh.
surface membrane. So a vesicle becomes docked and then fuses with the membrane. So this is a vesicle. This is the docked vesicle. This is a fused vesicle. So lots of cell physiology embedded in step 4. Step 5, what is step 5? Good, diffusion of neurotransmitter across the cleft, across the synaptic cleft. What else? Something else embedded in step 5. It's hard to see from the cartoon, but when neurotransmitter diffuses across the synaptic cleft, what does it do then? Binds to receptor, conformational change, receptor opens. So now if this is a ligand, if this receptor is a ligand-gated sodium permeable channel, that would be an EPSP. And if this is a ligand-gated, if the receptor is a ligand-gated calcium permeable channel, that would be an IPSP. If the receptor is coupled to a potassium channel somehow, and it opens potassium channels, it would be an IPSP. So it depends on the receptor. So we're just being generic here, but this is what triggers the EPSP or the IPSP that we talked about on Tuesday. And then what's number 6 supposed to be? What do you think happens? 6 doesn't say anything. After 5, what do you, and this is not something we covered in self-is, what do you suppose must happen after this, after 5? 8.
Well, you're gonna have a bunch more ion channels open, allowing for another depolarization of the next. I mean, you're propagating that action potential, right?
Oh, so we are,
Are you sorry? We're on.
we're not on the axon. We're way out on a dendrite here. So I didn't specify that, but we're way out on a dendrite. We are not going to trigger an action potential with this one synapse.
It's hard to tell where.
Yeah, so what's gonna happen at this location? Alice? Yeah, we have to clear. Remember, once we release and allow this synaptic cleft to be full of neurotransmitter, we've got to get rid of it for the next event. So so 6 is going to be removal, clearance of neurotransmitter. Eden?
So then, how is that this signal transmitted down the neurons so it can create an action?
So this EPSP would summate with the thousands of other EPSPs, diffuse down the dendrite
So you still have.
to the axon initiation, to the axon initial zone, and then at the axon axon initial zone, then the decision is made whether to fire action potentials or not.
But you still have a depolarization here.
Oh, we have a depolarization here. Oh, for sure. Or a hyperpolarization, depending on whether it's an EPSP or an IPSP. Yeah, that goes without saying. EPSPs are depolarization. Well, no, it doesn't go without saying. Let's say it. The EPSP is a depolarization, the IPSP is a hyperpolarization. So you should have decorated your slide with something that looks like this. Question, yes?
In regards to the voltage gated calcium channels, do you need to know anything about that being closed after the reactivity of
Do you have to know that the receptors close when the neurotransmitter is removed?
like about the calcium channels?
Okay, I could have said that that's pretty self-evident, but let's say it. If the neurotransmitter is removed, the receptors let go of their neurotransmitter and they are no longer activated, so they close. Yeah, that's a valid point. And just because I know it's true doesn't mean I shouldn't say it's true. It's true. Yes, question?
So they go directly from open to closed activity?
You mean these receptors?
No, the calcium.
Oh, the calcium channels.
Do they-
Wait, was that your question?
Yeah, that was my question.
Oh, I'm sorry, I thought you were talking about the receptors.
No, I was talking about the calcium
The
channels.
calcium channels go from open to inactivated and back to, and then have to recover from inactivation just like voltage-gated sodium channels. I'm sorry, Tyreek, I misunderstood your question. Voltage-gated calcium channels are like sodium channels in that they inactivate. And they have to recover from inactivation to allow this step to happen again. Receptors open in response to binding of the neurotransmitter and recover back to closed in response to the clearance of the neurotransmitter. And in fact, they have something like an inactivated state that is called the desensitized state, which we're not going to talk too much about. Thank you for your question. John?
Are calcium channels mainly just expressed at the terminal?
Calcium in humans, voltage-gated calcium channels are expressed in the nervous system. Calcium channels are expressed, not only, no. We have calcium channels expressed also in dendrites, so even though they don't fire action potentials in dendrites, they do participate in sort of the charge movement in dendrites. The ones we're talking about are highly expressed in the presynaptic terminal, and they are not expressed hardly at all along the axon, but they are expressed at the soma and in the dendrite, at lower levels, much lower levels. So here's what I wrote before, diffusion of neurotransmitter, but actually clearance would be a better word. Removal of neurotransmitter. So here is a cartoon of the snare complex. Some of you, I think, saw this in CellPhys. Here's the vesicle containing the neurotransmitter. Here's the membrane to which the vesicle is going to be fused. This is the synaptic cleft space here. Here are the V snares and T snare complex, this coiled coil alpha helical complex. And up there is the calcium sensor that binds calcium, triggers the conformational change. So here's a cartoonish, more cartoonish version. V snare, T snare, synaptotagmin, calcium channel, docking, coiled coil formation of the T snare, V snare, synaptotagmin complex, calcium influx through the voltage-gated calcium channels because of the invading depolarization from the action potential. Calcium triggers a conformational change of the synaptotagmin and the snare complex, pulling this conformational change sort of tightens the coiled coil complex and brings this membrane in contact with this membrane, causing it to fuse and dumping the contents of the vesicle into the synaptic space.
So it's this complex, the synaptotagmin and the snares are what cause the dock, not the RAB protein.
So there are other proteins that are associated with docking that this cartoon doesn't. There are RAB proteins that are also, there's getting rid of clathrin. There's a bunch of process that this cartoon doesn't cover. But those are also true. RAB is in here somewhere. And just as a point of interest, I brought in the tetrodotoxin from the fugu last time, and I brought in the trachotoxin from the poison dart frogs last time. Well, botulinum toxin is a toxin that paralyzes people who are infected with the botulinum bacterium, and it paralyzes people not because it changes channels, but because it destroys the snare complex. So there's a bunch of botulinum toxins that cut. They're proteases, and they specifically cut T snares or V snares and prevent the release of neurotransmitter by cutting the snare complex and preventing neurotransmitter release. And so in the absence of neurotransmitter release, we become paralyzed in the presence of botulinum toxin. And that has been medicalized to, botulinum toxin has been medicalized to make Botox. This will not be on a test. This is just for interest. So now let's talk about clearance. I want to come back to some of the concepts that we covered at the very beginning of the semester. And we're still sort of at the beginning of the semester, but in the first week. So does someone presynaptic terminal? Synaptic vesicles. Let's say this is this synapse is not releasing neurotransmitter. So some are docked and some are not. But neurotransmitter has been released into the synaptic cleft. And it has interacted with receptors. It's already done its thing, right? It's activated IPSPs or EPSPs in the postsynaptic neuron. Now we got to get rid of the neurotransmitter. What are some of the mechanisms for reuptake? Removal of the neurotransmitter from the synaptic cleft so that it can get back into synaptic vesicles where it is concentrated and stored. Can someone remember? There's a protein here. Wait, this is way out of escape. Okay, some of those words are right, but not all of them. What ion gradient exists across this membrane? There's more than one correct answer. What are the ion gradients that exist across this membrane? The sodium and the potassium gradient. Remember, the sodium potassium ATPase created a situation with a high concentration of sodium on the outside and a lower concentration of sodium on the inside. You used this during the problems. There are sodium neurotransmitter co-transporters that pump neurotransmitter. Oh, so the neurotransmitter is supposed to be green. So you can read that. The neurotransmitter is supposed to be green. Pump neurotransmitter into the cytoplasm. Now, I'm going to make a super big vesicle. How did we get the sodium gradient? There's a protein here that's key to this that is now invisible. The sodium potassium ATPase, the sodium pump. Just, it's there. You just have to know it's there, even if I don't cartoon it there. This is a synaptic vesicle, blown up for clarity. Two proteins that we talked about at the beginning of the semester. One that I cartoon like this, and one that I'm going to cartoon like this. What is this one? A hydrogen pump. A ATPase a primary active transporter that pumps protons into the vesicle. So where is the high pH? The high pH is in the cytosol. The low pH, remember pH, high concentration of protons makes for low pH. Just remember pH grows with this curve. High concentration of protons in the synaptic vesicles. Okay, we're not there yet. What is this protein? It's a neurotransmitter 1. Okay, it's a neurotransmitter 1. It's neurotransmitter, okay? Which way do I draw the arrow? Into, it's a neurotransmitter transporter, but it's a pump. Is there an ATPase function in this thing? No. What is it using? It's using the electrochemical gradient of protons. Protons are going down their concentration gradient and their electrochemical gradient. This is an antiporter that pumps neurotransmitter into the vesicle by using the proton electrochemical gradient as its source of energy. So this is reuptake and packaging of neurotransmitter. Now vesicles are also recycled, but we talked about that in cell phys. Remember endocytosis, clathrin coating, all of that cell physiology for the endocytosis. Remember, after a vesicle has fused, it goes through this process, and that's endocytosis. So the vesicle reforms by clathrin-coated endocytosis, and then the vesicle, which is still empty, gets refilled with neurotransmitter by this process of uptake and packaging. Laura?
Did you say that the neurotransmitter proton antiporter gets the energy from the proton gradient?
Yes. The energy for the neurotransmitter antiporter comes from the proton gradient. The proton gradient comes from the proton pump. Maybe?
What are the specific names for these pumps again?
This is a proton pump. Different neurotransmitter, so remember we've got dozens of neurotransmitters, and each of them has a different name, which I am not asking you to memorize. But if you want to know, let's go to Moodle. Let's look at key concepts. Here are the names. You want to know the names of the sodium-dependent co-transporters in the surface membrane. Here they are. SERT, serotonin reuptake transporter, is the serotonin transporter. DAT, dopamine transporter. NET, norepinephrine transporter. Choline transporter. GABA transporter for an inhibitory neurotransmitter. Excitatory amino acid transporter for glutamate and other excitatory amino acids. So that each one of the neurotransmitters has its own sodium-dependent transporter and its own vesicular proton antiporter. I'm not asking you to memorize these names, but they're there as a reference. Just for your interest, SERT is the target of the selective serotonin reuptake inhibitors, SSRIs. So this is the target of the antidepressant drugs of the SSRI class. Okay. So now, I want to finish with one more concept, which is presynaptic modulation. Modulation of the presynaptic terminal. So here's a cartoon of a presynaptic terminal. Mitochondria because it needs ATP. Vesicles, some of which have docked, some of which have fused to release their neurotransmitter. Neurotransmitter receptors, just the way we've talked about it. Two concepts. One is that the presynaptic terminal, in many cases, has receptors. We don't talk about them very much. We didn't talk about them in self-is, but now here we are in AMP, and I do want to talk about them. There are autoreceptors. The presynaptic terminal has receptors to the neurotransmitter that it releases. So it detects its own neurotransmitter, and those are called autoreceptors. They're not, this is not a transporter, this is a receptor. So these receptors will respond to the neurotransmitter and cause changes here. This is the autoreceptor is not the sodium-dependent neurotransmitter transporter. This is a different protein. Eden, I'll have to take your question after class. The autoreceptors are receptors to the neurotransmitter, but instead of being expressed on the postsynaptic membrane, they're expressed in the presynaptic, on the presynaptic terminal. And they modulate the presynaptic terminal by the neurotransmitter that it releases. That's what makes it an autoreceptor. In your coloring book, I think you were asked to color, but in any case, I'm going to mention it now, axonic synapses. Here is a axodendritic synapse, a classic synapse, like an axon making contact with a dendrite, with a postsynaptic membrane. But here is an axonic synapse. There are receptors on the presynaptic terminal that respond to a different neurotransmitter that is released by the this is a different neuron than this one. So we have 3 neurons here, 1, 2, 3. Axonic synapses can very strongly modulate the release of neurotransmitter at the axodendritic synapse of that synapse. This is presynaptic modulation of this synapse. So you can say, well, this membrane is postsynaptic, and this membrane is postsynaptic, and now we're going to get into some semantics. This is the main presynaptic, this is the main thing, and this is the modulatory thing. Axonic synapses are almost always modulating axodendritic synapses. So as homework, so I know you've got a quiz, but because we've run out of time, this would be a good question to review for the exam, and we'll come back to this next week. The presynaptic receptor at this synapse, so this is the modulatory receptor. The presynaptic receptor at this synapse is a nicotinic receptor. Would that increase or decrease the release of neurotransmitter from this synapse? That's a challenge question. The autoreceptor at this synapse is a G protein coupled receptor that is coupled to GI that activates K chains. Does the autoreceptor amplify or inhibit neurotransmitter release? I want you to think about those questions. They're simple when you get them figured out, but they're not simple until you figure them out. So, and then we'll come back to this on Tuesday, and we'll also start to talk about the wide variety of neurotransmitters.